Okay, so let's take a quick walk through classification, the lecture on classification of life. So this should be hopefully a very basic review of classification for most of you guys. I'm going to just talk briefly about classification process and some of the key things. I'm not going into detail on every level of classification of all the kingdoms, phylums, etc. in this lecture. As we go through the different units and the different topics and the different chapters, that's where we'll get into a little bit more detail about, well, why do these fish go into this phylum, or why are these microorganisms classified in this particular kingdom. That's where we'll get into a little bit more of the specifics. So this lecture is just general overview. There's a bunch of slides with information that I would use as a reference when you get into those further chapters. Bounce back and you might need to reference some of the stuff in this lecture. So try to tie them together. So, alright, so basic classification. Currently, scientists, we know of about 1.8 million species. That number's growing, constantly growing. Estimations I've seen 10 to 100 million species on the planet. I guess that's kind of the ballpark we think is out there. There's still a lot of stuff to be discovered, especially in the marine ecosystem. There's still a ton of organisms that we have no idea about. Same thing in tropical rainforests. Lots of things we don't know exist. It takes scientists to find those things. So when we're working with classification, we use a field called taxonomy. Now, taxonomy is the science of classifying an organism. And you're doing it based on physical features. Okay, what does it look like? Does it have 4 legs? Does it have this long of a tail? Does it have how many fins and what type of bone structure, etc.? That's taxonomy. That's how we're classifying a lot of things currently. Molecular information, DNA analysis, is changing this. So where we may start doing more taxonomy and classification based on a genetic analysis, we simply examine the DNA code, and that's how we organize and classify things. So once you've classified it, then we got to figure out how it connects, and that's what systematics. Systematics is the science of determining the evolutionary relationship between species. So how are they connected evolutionarily? What's the relatedness? Do they have a common ancestor in the recent past, or is it very, very far back before we see a common ancestor? So that's what systematics does for us, is it puts these things in a perspective when we're comparing them to each other and to the diversity of life, that 1.8 million species on the Earth. Now, when we use classification, we are using a system called the binomial classification system. This is how we keep stuff organized. The system is composed of a bunch of different levels. The key levels are going to be the genus and specific epithet, or species level. So the key level, when we give a species a name, and we use a name for it, we use the genus name, and then the specific epithet, or the species name. Alright, so the example I have here is from the rainforest. Iguana, iguana. That is very, very specific to that particular animal right there. Members of its population and similar animals just like that. So when you see those names, and scientific names are always written in italics or they're underlined, you know you're referring to a scientific name, not a common name. That's the problem, common names. We call that an iguana. Somebody else will call it bamboo chicken. Somebody else will call it big scaly lizard. You don't know exactly what you're talking about. So we want to try to get more familiar with the scientific names when we're talking about different species. Right? So when classification was being established, we follow certain rules. examples. I'm not going to read all this to you. I'll let you guys take 2 minutes, come back, read it. So pause the video, read it, and then come back. Okay, so you paused, you read it, now you're back. All right, so again, keep in mind, the name is in italics or it's underlined because it's based in Latin. You got the genus name, and then you have the species name or specific epithet. So Carol Linnaeus is one of the founders of the scientific naming system. So we call it the Linnaean system. When we look at these things, we're still using a lot of his original work with creating the names. Latin was primarily used or is used because, you know what, that's the language that was spoken when this was first started. It's also still used because, for the most part, Latin is, I hate to call it a dead language, but not a very common language. So it makes sure that it retains the integrity of the names when we use a language that is not constantly changing and adjusting, etc. All right, so we want to keep that in mind when we're naming organisms. So when you see a name or an initial following a scientific name, that indicates the author or who named it. So the authority, the authority of the naming individual is abbreviated, or just using a little L for Linnaean, Linnaeus there. So cool thing, you guys find something brand new to science during this trip, during any trip, any research, anything, you get to name it. You get to name it. Usually you're naming it at the species level, but maybe you find a brand new genus. That opportunity does exist when you get into the marine world, because there's still so much we don't know about it. Sometimes, though, you're going to get, oh, wow, I got this organism that fits into the genus, it fits into the species, but it's just a little bit different. And that's where we might create a subspecies category. And that's where we get into a third name, what we call the trinomial nomenclature, a third name for that organism. So an example for us, Homo sapiens sapiens. That makes us a little bit different than the earlier Homo sapiens that existed on Earth 25,000 years ago or 50,000 years ago. That time frame makes us slightly different, putting us into a subspecies category. All right, so the big things we want to look at with naming is, how do we start out? Well, when we start naming, we need to place the organism into a domain. Currently, we have 3 domains recognized by the scientific community. Domain Eukarya, we have domain Archaea, and we have domain Bacteria. Now you'll see different derivatives of this. It might be domain Eubacteria, domain Archaea bacteria. Subtle differences, but those are the 3 recognized domains in the scientific world. Within the domains, we then have 6 recognized kingdoms at this point, currently. So underneath Eukarya, we have kingdom Animalia, all the animals. We have kingdom Plantae, all of the plants. We have kingdom Fungi, all fungus. And I'm going to put a little asterisk here because this is a goofy one. Protista. Protista, by and large, is not recognized as a kingdom. It's being reorganized and readjusted. So it's creating a little bit of a kind of a stomach ache for a lot of people. But for now, we're just going to consider those 4 sitting under Eukarya. This could change in the near future. Protista is changing, but for now we're just going to keep it basic and say, all right, let's focus on those 4 under Eukarya. Within the domain Archaea, we have the kingdom Archaea. So that makes it easy. That's also being explored and studied, and proposals for reorganization. Within the domain Bacteria, right now. Kingdom bacteria, another one that's being proposed and discussed and expected to change in the near future. So there's a lot happening with classification. I'm not going to ask you guys to get too crazy and overboard on classification. We're going to keep it kind of a broad general picture because so much of this is changing. Again, as we go into the different lectures, we'll talk about the unicellular organisms in the ocean. We'll talk about the animals with backbones, animals without backbones. That's where we're going to get into the details of why does it go into this kingdom or this phylum or this class or this order, etc. So get comfortable with that hierarchical system at the bottom here, the domain, kingdom, phylum, etc. Make sure you're comfortable with how that works. Domain, kingdom, phylum. If we're talking plants, we slip in that word division. Then we go to class, order, family, genus, species. Okay, so like I said, each lecture over a different group of organisms, that's where we'll get into those details about each of those. But for now, what I want you guys to do is fill out how we would classify humans. Starting at the domain, work down to kingdom, phylum, class, order, family, genus, species. How would we classify those? What domain do we go into? What kingdom, what phylum, etc.? Fill it out. Make sure you guys fill it out. I'm trying to give you the big hint. Fill it out so you know what the classification is for us. Alright, so another one of the slides, I'm not going to read all this to you, just more details about how we classify, kind of the history. We used to have this old 5-kingdom system that got changed. Now we're at the 3 domains. Again, a lot of this is changing. Get the fundamentals down about classification. What are the key things? And you guys will be fine. Okay? Alright, so when we look at that survey of different kingdoms, what I want you to be comfortable with is at the kingdom level, what are the key features? What makes something go into archaea? Single-celled. These guys exist in extreme environments. Hot, 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 nasty, salty, phosphorus, methane, etc. Tough environments. But we're also finding out these guys can be all over the place as well in environments that we didn't expect them to be in. But a big thing with the archaea is when you're looking at them and studying them, you look at their cell wall. They don't have this structure called peptidoglycan. That's one of the things that we want to keep in mind when we're looking at archaea versus the bacteria, or eubacteria as sometimes they're called. Because the eubacteria are also single-celled. Wide range of environments. They have a cell wall that contains peptidoglycan. So that's going to be a big feature for the bacteria. We'll also talk about this thing called the nucleoid region in the bacteria. Houses all of their DNA. Again, we get into a further lecture, we're going to talk about bacteria and archaea a little bit more detail. I'll have additional things for you guys to look at in regards to their key traits and their key features. Okay, Kingdom Protista. I'm just going to call these guys a mixed bag of organisms. They basically don't fit in plants, animals, or fungi. That's why they're here. So we'll get into some examples, talk about, oh, it's got this trait, that trait, etc. This is why Protista is being reorganized currently. Okay, so that's our first part in this lecture over classification. Pop open the video for the next one. We'll start talking a bit more about some of these other kingdoms, just brief highlights,